So today we are now on session 2. Sorry, this is not the right slide. Let me go back here. Okay. All right. So again, let's begin our session. This time we're now on session 2. This time we'll focus on Mendelian genetics. So I believe in grade 8 you've actually had the introduction or the experience on how to do or how to solve problem solving related to Mendelian genetics. Okay? So, this is actually found on our book, pages 63 to 65 under chapter 3. So, just a quick recap regarding our activity yesterday. So, we had our first session, and we had majority of our session was actually allotted for the making of the trait bingo. And after that, we had actually our processing or the summary of our discussion or our activity. We were able to point out that there are observable characteristics, and these are actually what we call as traits. And when you talk about traits, aside from being observable, they are also inherited from parents to the offspring. Now, a certain trait will have multiple variations or options. And lastly, one important thing that we've learned about um, genetics is that in a given trait, there are usually at least two options. One, we have a dominant allele, and the other one is the recessive allele. So now that we have discussed that, please take note. Um, the module is already available on our CLEAD. This will cover 2 weeks. Okay, so this week and the week 2 for week B. What is our objective for this afternoon? We only have one, and that is to explain the concept behind dominance and recessiveness. So if you remember, when you talk about trait, especially when you talk about Mendelian genetics, it's very crucial that we understand the effect and, of course, the relationship between dominant allele and recessive allele. So we will revisit that later on. So I have here a very simple question. Do you still remember the father of genetics based from your experience from grade 8? Anyone? You can mention his family name, father of modern genetics.
Record Mendel.
Okay, Gregor Han Mendel or simply Mendel, right? So, of course, probably you have seen his face in some of your PowerPoint presentation last year or some. Okay, thank you, Miles, for informing me. Okay, so what happens is that, of course, you have probably seen or encountered at least his face. Now, looking at his picture, what do you think is his profession? So, of course, this picture is a cartoonized version of him. But based on what you can see or observe on that given picture, what do you think is his profession? Anyone?
Pare?
Pare. Okay, very good. It's a person who works for the church, or we can say a priest, a monk, or a person who is religious, right? Now, aside from that, what do you notice on his hand or what he's holding? Do you recognize that particular plant? Any idea for the name of that plant? Okay, pea plant, correct. So, it's very important that we understand why I'm asking this question is that Gregor Mendel is actually, first and foremost, his profession is actually a monk or a priest. But because of that, his work, he was actually um, sent to study further his education for his work as a priest or a monk. And when he returned to his monastery, it is then he developed or he had this um, interest towards growing plants or specifically pea plants. So there is a video on our module that will discuss on us briefly the life and work of Grigor Mendel. Okay, so what I want to emphasize here, so you can watch actually the video during your most convenient time. That's only 2 minutes and 35 seconds. So what I want to draw or emphasize here is that Grigor Mendel is a priest, a person who is religious. At the same time, he has interest in science. So as you can see here, religion and science can actually work together because initially during probably you've heard there are many conflicts and resistance between the two groups, the group who are considered to be scientists and those who are religious. But here Grigor Mendel is a living proof that science and religion can actually work hand in hand and work together. Okay? So again, I am encourage you to please watch that very short video regarding Grigor Mendel's life and his work. So again, he is quoted as the father of modern genetics because he actually paved the way or started the foundation for what we know about genetics and heredity. So here again, as I've mentioned, he is initially a Augustinian friar or monk or a priest, but he is also interested in meteorology, mathematics, botany, biology. And of course, he is well known because of his work on breeding pea plants, and which actually gave him the recognition about being the father of genetics. However, his work was not easily or readily recognized during his time. It was actually later after his death that his work was revisited by other scientists, and they actually recognize his contribution to genetics and heredity. So, what are the most important contribution of Mendel to genetics? Number one, we have, of course, the dominant and recessive traits in pea plants. So he was able to distinguish or um, uh, just distinguish or make a list of which of these traits are considered to be dominant or which are the alleles for that particular trait. So he was able to propose that there are actually 7 traits observable on a pea plant. And for each particular trait, there are actually 2 options or alleles. So example, you have seed shape, seed color, pod shape, pod color, flower location, and plant size. And for each trait, example, you have seed shape, your seed or the seed of the pea plant can either be expressed in a form or observed as round or wrinkled. And because of his repeated tests and breeding of this plant, he was able to propose that there are certain alleles that are considered to be or classified as dominant allele, while others are identified as recessive allele. So when you say dominant, it's actually based on his definition, these are the traits that are repeatedly or more prevalent than the other option. Another contribution of Mendel to genetics is that he was able to establish the concept of phenotype and genotype. Of course, during this time, the understanding about DNA and the genetic material is not yet really understood, but he was able to um, present the concept using certain system. So he was able to establish that when you talk about characteristic, they can be expressed in 2 ways. We have a phenotype or a genotype. Now, here is an example or a comparison between the two. One on the left side is the genotype, and on the right side you have your phenotype. So I believe these terms you have encountered them in your grade 8. Am I right? So let us differentiate them. So when you talk about phenotypes, it is actually the description of the observable characteristic that is observed or seen on the organism. Usually, it talks about the morphological or the physical form or the structure of the organism. Example, it talks about the color, the texture, the length, and so on. So, phenotypes are written in description form or using descriptive words. However, on the other hand, when you talk about genotype, geno means genes. So here, of course, as I've mentioned, gene is not readily understood during his time, but he was able to propose a system wherein we can the code as particular characteristic using letters. Okay? So when you talk about phenotype, you are expected to see that they are expressed as letters and codes. And later on, we will explore or revisit, because again, you've learned this from your grade 8, how or which letter to choose in terms of the letter coding. Okay, so example, let us say we know or it has been established that yellow seed is dominant over green seed. So you have here homozygous dominant yellow seed, heterozygous yellow seed, homozygous recessive green seed. Is this an example of a phenotype or a genotype based on numbers 1, 2, and 3? How do you describe this given here? Is this a phenotype or a genotype? Anyone? Hello? Is there anybody there? Homogeneous. Yes, anyone? Is this an example of a phenotype or is this an example of a genotype based on what I just mentioned a while ago in terms of their difference?
Phenotype, Chair.
Very good, Ian. So this is an example of a phenotype. All right. Again, when you talk about phenotype, it's descriptive or it's a description of a characteristic. All right. So later on, we'll understand that better. What do we mean by homozygous, heterozygous, and so on? So, what about if you encounter this as your example? You have capital Y, capital Y, and small letter y. Is this an example of a genotype or a phenotype?
Genotype.
Very good. So this time, this example here is actually a sample or an example of a genotype. Notice that here, we're no longer dealing with words. Instead, we're only using two letters, and they are actually the same letter, and they are just different in terms of their cases or capitalization. So take note, when we write the genotype, they are always written as a pair, okay? Because each letter represent a code or a gene from the parental side or the father side, and the other one or the other letter is from the mother side or the maternal side. So when you're asked for a genotype, it is always written as a pair, right? Hopefully that's clear to everyone. Now take note, for each corresponding phenotype, there is a corresponding genotype and vice versa. Let me repeat. For every phenotype, there is a corresponding genotype and vice versa. So what does it mean? It means that if there is a phenotype, you can actually write its corresponding genotype. And if there is a given genotype, you can actually describe the corresponding phenotype. So that's phenotype and genotype. This time, let's recap or recall the difference between the two concepts, trait and allele. So when you talk about trait, I think I mentioned this yesterday. A trait is the observable characteristic. Usually, a trait is physical. or morphological, meaning you can observe it directly. But however, it's not limited to that. There are other traits that are also not ob- readily observable on the organism, like attitude, preferences, and so on. Intelligence. You don't see that, but of course, if the behavior or if there's a stimulus, you can actually observe that. So going back to the trait. So example, you have here seed color, hairline. These are the traits we actually use during our trait bingo yesterday. Now, how is this related to allele? Remember, a trait is the observable characteristic. But when you talk about allele, it is the variation or the option for that trait. So let's take an example, seed color. Now, according to what Mendel proposed or was able to um, conclude in his observation, that for a seed color as a trait, the available option for that are either yellow or green. Okay, is that clear to everyone? Trait is the characteristic, or we can say that it's a category, and then the options or the available variations of that trait is generally, according to Mendelian genetics, at least two. You have two options. Later on, when we proceed to non-Mendelian, we'll learn that the options for a particular trait is not limited only for two. Okay? Is that clear to everyone? Are we clear on the difference between the trait and allele? Questions between the trait and allele? Okay, wala. Everybody is silent. Okay, we have here, let's now try if we really understood or remember now our concept of dominance and recessiveness. So, again, allele is the option. So let us have here our given situation or scenario. So given, you have here the given allele and the corresponding code. So if you remember, sa Mendelian genetics, what we usually use as our letter or the letter that we will use for the coding is the first letter of the dominant allele or the dominant option. So in this case, yellow is dominant over green. So what's the first letter of yellow? Anyone? What's the first letter of yellow?
Capital Y.
Y. So that means using that letter, the capital of that letter will go to the dominant, and the lowercase or the small letter for that letter, the same letter, will go to the recessive allele. Same letter, they differ only in terms of their capitalization or cases. Claro? Is that clear? How we got Y capital and Y small for dominant and recessive respectively? Okay. Yes. Okay. Thank you for that, Aliana. So that means using that coding, we will now give the corresponding um, what you call is phenotype and genotype. So let us say if your first given is homozygous dominant yellow. So when you talk about homozygous, it actually pertains to the first um it's a combination wherein the letters are of the same cases. So you'll notice since you have a dominant yellow, the corresponding genotype is capital Y, and that is described as homozygous dominant yellow. Two capital letters. Now, what if your given is homozygous recessive green? So in this case, you have the same letter Y, but this time both letters are in lower cases because we are now talking about recessive. Okay? So we have two small letter Y. So same case in this time in this scenario, your letter or the capitalization of your letter is in lower cases, or we know it as small letter. Now, The third possible option is, or possible expression of this um, characteristic is we have the term heterozygous, meaning it's a combination of a capital and small. So they differ now in terms of their cases. One is capital letter and the other one is small letter or lowercase, which means that the yellow um, overshadow or hide the presence of your green allele in your genotype or your genetic code. So let's try if you understood this one part. So we have here, our scenario is this time we're talking about the seed shape. Okay. One is round, which is the dominant, while wrinkled is the recessive. So how do we write the genotype? Now, first of all, should be the first letter, or what's the first letter of our dominant allele?
R.
R. That represents actually round. So that means when we code or get the genotype, they differ in terms of a capital R or a small r. However, let's start with the first example. You have here homozygous round shape, all capital R or all small letter R, since it's a homozygous, which is the correct option. Okay, very good, Sophia. So it should be capital R, capital R. Now, what about if you have wrinkled? This is the recessive. Capital R, or small r. Okay, very good. So this is now recessive. Okay, remember? The same letter, but they differ only in their capitalization or cases of the letter. Now, what about the third question? You have here a heterozygous. Very good, Miles, Sophia, and Rupert. So, hetero means you have now two cases, different cases or capitalization. One is capital and the other one is small letter. All right. Okay, let's really check if you understood. This time, we're talking about the length of the stem of the plant. What is the dominant allele? Long or short? Based on the sentence here or based on the scenario?
Long.
Long. So what's the first letter for our dominant?
L.
L. So that means our coding will use either capital L or small l. Now this time, let's do the reverse. You're given the genotype. You are asked to give the corresponding phenotype, meaning the description. So here on the column on the right side, you have capital L, capital L. Homozygous or heterozygous?
Homozygous.
Yes, homozygous, same capitalization. But is that dominant or is that recessive?
Dominant.
Dominant. So what's the characteristic of a dominant? Short or long?
Long.
Long. So if we put that together, this is described as correct. Very good. So we have Jade and Miles. So it depends on you if you want to write it in a complete manner or very, very detailed manner, or you can have a shortcut. So you have here homozygous dominant long stem or simply homozygous long stem. Okay, how about the second example? This is small letter L, small letter L. Who wants to try? If you want, you can also write your answer on our comment section. Yes, Sophia, we have here homozygous short, okay, stem. So the stem is here STEM. So we have, okay, correct. So it's either you write it as homozygous recessive short stem or homozygous short stem. Now, how about the last example here? We have L capital and then we have a small letter L. Okay, very good, Rupert. This is heterozygous. But instead of writing two, long and short, you can only write the dominant, which is heterozygous long stem. Okay, I hope it's clear to everyone how to write the phenotype and genotype respectively. All right? So let's go to the next. So more or less, we are now ready for the exercise. So another video reference is available um, on our module. So I highly encourage you to watch this during your most convenient time. Okay? So it talks about dominant and recessive. So I believe I already explained this one. So let's try um, using the concept of Mendelian genetics in terms of answering certain problem set. Now, the problem set is actually available also on our module, so you can actually uh, revisit it later on. Okay, so let's talk about first the description of the problem. In this case, it talks about the shape of the pea plant. One is spherical and the other one is dented. In other books, dented is also described as wrinkled. Okay? And sometimes some other books they use instead of using the word spherical, they use the word or the description round. So, spherical is dominant over the dented characteristic or the dented option. Now, two plants that are described as heterozygous for the seed shape were asked to pollinate each other or breed or do the reproduction. So we are asked to answer the following. What are the possible answer? The first one, what is the genetic code or the genotype for our parents who are both heterozygous? And then the second is that we are asked to cross the two parents so that we can identify the possible genotypes and phenotypes of their offspring. And lastly, we are asked about the probability or the chance if if there is a chance or a probability that one of their offspring will be heterozygous spherical seed or will have heterozygous spherical seed. So let's start. Okay, so since this is the beginning, I believe you all actually had this in grade 8. So just to rejig our memory. So the first question is, what are the genotypes of the parent? So here, so that you can visualize, one is wrinkled and the other, or one is dented and the other one is spherical. Now, which do you think is the dominant option here? The one on the left or the one on the right?
The one on the right.
Okay, right. So it's spherical or round or it has a smooth surface. Wala siya mga dent sa iyahang side. So that means this is more prevalent, balik-balik siya, mas obvious siya sa um, group of pea plants or a population of pea plants. So, based on the description, both parents are heterozygous. But let's talk about the trait. The trait talks about seed shape. And the options are between spherical and dented. So how do we write the code for two heterozygous parents? Is it capital S, capital S, small s, or S small, S small? They are both heterozygous. What should be their coding? Again, my options are capital S, small s, or small s.
Capital S, small s.
Okay, very good. Since both parents are heterozygous, that means they are both carrier of spherical and dented. However, what is seen or expressed on the organism is simply the spherical characteristic. So, of course, probably you've encountered this before. Punnett square. Do you know what's Punnett square? Ano man ang Punnett square?
Para
Punnett, ay, Punnett square. No, correct. Okay, continue, Miles. I was
ay
just really
kadang
fast.
kwan, sir, para ma-calculate ang probability sa traits,
Yes,
sir.
very good. So this is actually a technique that will help us determine the probability as mentioned by Miles if we combine the genes of these parents. So take note, both parents are heterozygous. Now, since letter S, okay, it's really difficult to identify if it's a capital or small. What I did is the capital letters, I highlighted them in red, and the other one, the small letter, is in black. So let's distribute that here. So one parent, one side will be, let us say, parent one or father plant, and the other one is for the mother, or let us say parent number two. Now, what do you think is the result on the first quadrant here? We have S capital and S capital. So that's S capital, S capital. Okay, by the way, it should be red here. Kani red ni siya. Okay? Okay, let me, sorry ha. Kuan ko, O si gamay. Okay, let me turn that red. Okay. Okay, and also this one. Okay, here. So here we have the first quadrant. First quadrant, we have capital S, and then on the second quadrant, okay, I hope you can see the movement or the combination. And then lastly, and then of course, this one. Now, of course, as I've mentioned, the phenotype is, or the Punnett square, or what was mentioned by Miles, this is only the tool that allows us to predict the probability. But of course, at the end of the day, it's very important that we understand what these letters mean. So what are the phenotypes of our offspring? We have, on the first, we have homozygous spherical, that's capital S, capital S. Heterozygous spherical, that's capital S and small letter s. And then lastly, on the fourth quadrant, we have small s, small s. So here are also the genotypes. Now, looking at the probability, you have four quadrants, right? Or four squares. So looking at this, how many out of the four squares actually are, what do you call this? Actually houses the heterozygous spherical capital S, small s. How many out of the four boxes or squares on the Punnett square? Pila man siya? So this one is capital, capital. So that's not included. This is capital and small. That's one. That's capital and small. That's another. Here, it's small, small. So how many in total out of the four squares? Okay, that's two. So 2 out of 4. Or we can simply say that's 1/2 or 50% chance or probability that the offspring will be heterozygous spherical or with heterozygous spherical seeds, capital S, small letter s. Okay, question before we go to problem set number 2? Any clarification? Wala? Hello? Wala. Okay, very quiet. So let's go to problem set number 2. So let me ask you first, based on what you can read on the statement, which allele or option is dominant? Short or long? Okay, very good.
Short.
It's short. That's why we will be using S rather than L. All right? So even if it's stated differently, the way we understand it, it's short is dominant while long is recessive. Now, it talks about, so we will encounter the same set of questions later, but this time we're talking about short tail and long tail. So probably those um, who have pets, especially kanina mga iring, they're very expensive, right? So sometimes they are classified or identified based on their, either you like short-haired or long-tailed cats. Okay. So let us say you have here so let's talk about first the trait. What's the trait here? What's the characteristic that we are talking about? It talks about the hair or the length of the hair of the cat. So our options are either short or long. Now, our male cat is homozygous short-haired, while our female cat is long-haired. So how do we write first the genotype or the code for a short-haired male? Homozygous man. So that means same capitalization. Short-haired man siya, so capital S, capital S. Very good. How about for mother or the female cat? She is long-haired. All capital S or all small s. Okay, very good. So just be careful with the capitalization, okay? So father cat, capital S, capital S. Mother cat, small s, small s. Okay? So here we have a father, the male cat, and the mother cat. So capital and small. So this time I will allow you to do the combination. So first quadrant here, who wants to try? This is capital and this is small. What would be the resulting genotype for this square? Okay.
Heterozygous, capital S, small
Heterozygous.
s.
Very good, Sophia, Ira, Miles, and Lawrence. How about the second? Is it the same or it's a different code? Okay, very good. So it's the same, capital S, capital S. You can actually say that all other four squares will actually have, all of them have a capital S, small s. So if you're asked for the phenotype, how many phenotypes do you think you will have here? Ang combination niya. Do you see other combinations on the Punnett square? Pila ka letter combination lang siya? Okay, one, since they are all uniform. So how do you describe that? Homozygous or heterozygous? Capital S and small s. Okay, heterozygous.
Hetero.
Very good, hetero. But is it short-haired or long-haired? Okay, short because again, this is with dominant trait. Very good. So all of them are described as heterozygous short tail or with short tail. How about the genotype? So all of them, they have uniform genetic combination. So all of them will have capital S, small s. Now the question is there a chance that you will have a homozygous long-haired cat or kitten?
No.
No. So none. So there's no chance or zero chance that any of the offspring will be homozygous long-haired. But what about if I remove homozygous? Let us say, is there a chance that the offspring will have long-haired? Is it the same question or na different na ang concept? If I remove homozygous, will the answer be different?
Yes.
Yes, sure.
So, what happens then? Zero pa Japon or naanay chance if I
Nanay
remove the homozygous?
chance.
Okay, what's the chance? Is it 0, 25, 50, or 100%?
100.
Yes, very good. Because all of them are heterozygous. Ay, sorry, long man. Kung gi-remove na ko ang homozygous long. So, dapat, wala gyapon siya chance. Kay, di ba? They are all short-haired. I'm just making you verify. Kay long is a recessive trait or sorry, recessive allele. So, even if I remove the homozygous there, there's no chance na na ay long-haired because all of them are actually short-tailed. All right. Last, this one about earlobes. So I'm not sure if you are particular or keen on observing the shape of your own earlobes. Okay, so we have here, we have an attached, and then the other one is free earlobe. So you'll notice na lang siya slight difference. Kaniyang tip here, naka-attach siya sa skin directly. But others, naa sila'y detachment, meaning na ay gap in between sa lobe and sa skin, the actual facial skin. Okay. So, your your woman is, or the mother here is heterozygous, while the father is a homozygous recessive. Okay? So, mother is heterozygous, father is homozygous. Father and then mother. What do you think will be the result here? Anyone who wants to try? The first quadrant. What could be the result? Okay, correct. That's capital F and small letter f. How about the second quadrant on the right side? Okay, it's also capital F, small f. So take note, remember, we're just simply combining them based on the quadrant. And then how about on the third? This one here, this quadrant. What could be the result? Very good, Ira, Jade, and Aliana together with Sophia. They are both small letter. So same thing here. Yes, Isa. So looking at the Punnett square, how many combinations do you see? One, two, or three? Okay, you only see two combination. Capital F, small f, and the second combination is small f, small f. So what are the genotype? How do you describe capital F, small f? Homo or hetero? Okay, hetero. How about if you have a small letter f, small letter f? Is it homo or hetero? Okay,
Homo.
homozygous. So let's start with the homozygous, that's recessive. That means it's attached. While the heterozygous is will have the free hanging or detached earlobe because again, we will or what is manifested on the organism is the dominant allele. Carrier lang siya sa recessive. Okay? So what are the combinations? As you mentioned, there are two possible combination. One is F capital F small and the other one is F small F small. Now the question here, is there a chance that you will have a heterozygous free hanging earlobe? Yes or no? Okay, very good. That's a 50% chance. So if you look at it, hetero, this one, it's look at the Punnett square. There are 2 out of 4. And if you do the math, if we divide that, that will actually give us 50% or half of the chance that the given offspring will have a free hanging earlobe. Okay? Question? Violent reaction? Okay. So take note, this is just a review of what you had in grade 8 because later on, we will improve on this concept regarding dominance and recessiveness. Okay? So if there are no more questions, it's now exactly 1:20. Okay? So again, please do not forget the things I mentioned about this one here. The first one about the letter survey. Please ask your parents to click this here and this will automatically redirect them to the Google form for the survey. And the other is about your verification of your name so that I can correctly encode your name in case there are corrections here or there is a lacking name from your from the one that's given from the registrar. Okay, so please take note and also if you have extension as part of your um, full name. All right, so take care everyone.